Thank you, Cindy. Thanks for the invite to come up here. What I'm going to talk about today is mealybug pests in California vineyards and orchards. Thank the co-authors, folks that have been working on these different insect pests, mostly in grapes. And I've spoken to you before about the vine mealybug. So while a lot of the information today will still be vine mealybug focused, that certainly is not the only mealybug pest in California. We've got a whole series of them. We've got mealybugs that are attacking palm fruits, mealybugs that are attacking citrus, mealybugs that are attacking pomegranates, mealybugs that are attacking pistachios. So what's unique about California, and I've mentioned this before, California really has been the dumping ground for the world's mealybugs that attack specifically vineyards, but other species as well. So while the vine mealybug has been getting so much attention, it's really only one of about 8 important invasive mealybug species in California. So vine mealybug, we know from work that I presented in the past, is Mediterranean in origin. We know our vine mealybug in California came from Israel, Egypt. I've worked with some European researchers, taxonomists, and now we've even separated out the vine mealybug from Israel and Egypt, compared with the vine mealybug from the rest of the Mediterranean region. So, for example, you might talk to someone from a vineyard region or in Italy or Spain or France, and they'll tell you, the vine mealybug is not really a major pest. We actually get more citrus mealybug. Well, that's because it's slightly different than the one we've got here. It is an invasive pest. We can see here some of the regions it's now found in. Throughout the Mexican vineyard growing region, California, it's now in Oregon. Temperature models say that it probably can go to Washington and the Okanagan Valley in Canada. It's in Brazil, it's in Argentina, it's probably in Chile, it's certainly in Peru, and it's in South Africa as well. Can you kill it? Can you eradicate this? Really, we tried. When it got to Napa, I worked with the county egg commissioner there, and from 2002 to 2005, they really tried to knock this pest out. In fact, we had insecticide programs when it was found. We were using a delayed dormant lure-span, bloom time spray with neonicotinoids, in-season spray with lanate, dimethoate, or imidan, and an IGR such as Applaud, and then a post-harvest treatment with lure-span. You can't do this anymore. It's not legal. But we were trying to do this, monitored for a minimum of 1 year after all these applications. And from 2002 to 2005, the population only continued to increase. So these are a hard group of insects, the mealybugs, to control, to wipe out. And this just shows this from California. It was spreading on leaves that were infested, on harvesting equipment. It got into the nurseries and was spreading on rootstock. Great talk about glassy-winged sharpshooter. During this 1998 to 2001 period, we were very worried about glassy-winged sharpshooter moving throughout the state. And so all of the nursery material was being sprayed with Pravado. That's imidacloprid in a foliar form, which is great for glassy-winged sharpshooter, but it doesn't control the vine mealybug. So from 2001 to 2008, vine mealybug really spread throughout California. I don't know that it's in Mendocino County yet. I suspect it is because, again, it's been found in Oregon as well. But as mentioned, this is not the only mealybug in California attacking orchards and vineyards. Citrus mealybug, probably native to Asia, found throughout the world, looks very, very similar to the vine mealybug. Both have got this short tail end section with waxy filaments all the way around. That's the head up there. It's a pest of citrus. In Europe, it's also a pest of grapes and vineyards. I haven't seen it in vineyards here yet in California, but it might be misdiagnosed as the vine mealybug because they look so similar. There's a whole group of these Pseudococcus mealybug species. The grape mealybug is the only native mealybug attacking vines that's found commonly on the leaf tissue. It's the one where if you poke it, it has this orange fluid coming out. It can be told apart from the vine mealybug and citrus mealybug, not only by its biology, but also because these tail filaments at the end, these are just waxy secretions, are much, much longer in the grape mealybug. A very close relative of the grape mealybug is the obscure mealybug. This is more of a cosmopolitan pest. When you poke it, this is the tail section, this is the head section, but it's got these osteolar pores all the way around it, as all these mealybugs do. When you poke it, it will come out with kind of a clear osteolar fluid. This is defensive secretion. It prevents some predators from attacking it, kind of annoys them, and they go away. Again, an invasive pest. When we look at its distribution, it probably is from, I think this one's from South America region, native to probably, the temperate region around Chile, Argentina, southern Brazil. But it's now, that's in green, it's now found in red really throughout all of the world's temperate regions. So obviously it's not in northern Canada and northern Russia, but just it's invasive in all these countries throughout the temperate region. This is a pest of a lot of ornamental plants, so it really was spreading on greenhouses. For us, it can also attack some of the palm fruits, so it's a great pest, but attacks palm fruits as well. For our vineyard regions, I really find it most commonly found in the Central Coast area, but I found it in Fresno, I found it in Lodi Woodbridge. I know it's been reported in Napa and Mendocino. And again, it is more common on ornamentals. So in glass houses, it's very common to find it there. Another Pseudococcus species, which is very common in glasshouse systems, is this long-tailed mealybug. It comes from Australia. For us, I've never really found this attacking many other crops except for vines. And in vineyards, it's really in the Central Coast area near Santa Maria. I don't even find this in Paso Robles. It doesn't do well in really warm climates. So it would be more likely to get into, it can attack apples, it can attack pears, it can attack vines, but most likely it would be moving from an ornamental, which is around your house someplace. And I just don't find this commonly attacking any of our crop systems. Again, the one exception is that I found it in Santa Maria in vineyards. Other Pseudococcus species include the Comstock mealybug. This one is from Japan and China. Like the other Pseudococcus, it's got a tail section, a little bit shorter than the great mealybug, or a little bit about the same size as the great mealybug, a little bit shorter than the long-tail mealybug. And we've also got the Citrophilus mealybug from Australia. And what's interesting to me is this can be a major pest in some regions of New Zealand on grapes, but I just don't find it on grapes in California. I found this down in Southern California. It can be a pest in apples and pears, but I think it's got some temperature intolerances for the colder climate as you go a little bit further north. Gill's mealybug, this has become a major issue in some pistachio and almond fields, although in the foothill areas around Placerville, this is a vineyard pest as well. To me, I can tell this apart from the Pseudococcus. This is the first Pharesia species. It produces these glass rods. And like the long-tailed mealybug, it doesn't have, it's not a true ovisac in that it pushes out live first instars. So the biology of these mealybugs we'll go over a little bit. They're all fairly similar, but this and the long-tail produces live first instars rather than crawlers. It's probably from southeastern US, closely related to the Brigada mealybug, which is from Jamaica. So here's just a list. I haven't talked about the ground mealybugs, which you can find sometimes on the roots. Very different in appearance, no real tail, no real waxy filaments. And the pink hibiscus mealybug, which is found in the southern California right now, attacking vines, but I not heard it making it into the San Joaquin Valley or further north. You can tell these insects apart fairly easily, but there's also been a multiplex PCR ID made for most of these species. So that can tell some of the first and second instars apart, which I can't even tell first instars apart from each other. And notice that on the identification, I oftentimes used this tail section. This is just a waxy secretion. So we see here is a just molted grape mealybug, and you can see there are no waxy filaments. It's got this pinkish, brownish color to it rather than this whitish color. Again, the white color, the tails, that's just from this waxy secretion. So it's not a good ID to separate vine mealybug out from the others all the time. And they all have got a fairly similar biology, except that, again, Gill's mealybug and long-tail mealybug push out first instars rather than the egg. We've got the male will have a prepupal stage and then a winged adult male. And that's over here for the male line, and for the female line, 3 different molts until they get to the adult female. I've found that most of these insects in the field have about 100 to 200 eggs per female, but in the laboratory, underneath good conditions in the field, as many as 750 eggs have been recorded from one female. Vine mealybug has more eggs than most. Citrus mealybug has quite a bit. Grape mealybug seems to have the fewest amount of eggs to me. And the numbers of generations differ amongst these pests, these different species as well. So for example, our native grape mealybug will have about 2 generations per year, with a summer egg development coming out, overwintering eggs, crawlers in 2 different periods, nymphs in 2 different periods. That's going to make it much easier to control because you've got a period in time with egg masses coming out, with eggs coming out, and then crawlers following that. So you've got these spray periods to apply your pesticides. Vine mealybug, in contrast, in your area, will have 4 to 5 generations per year, 6 to 7 generations per year in my area in Fresno, and maybe up to 8 generations where David Havlin is down in Bakersfield. So the more generations you've got, the faster the mealybug is feeding, the more it's putting out honeydew, the more crawlers you've got. And this means that you're going to have a better chance for movement between vineyard plots and for spread of diseases like pathogens. Again, we've got mealybugs attacking all different crops. We just see it here on pistachios, apples, grapes. One of the problems with mealybugs is that they can vector diseases. All mealybugs in vineyards that we've tested have been vectors of grape leaf roll-associated virus. So again, vine mealybug, more generations per year. That means more crawlers are present throughout the year. That makes it harder to control. And crawlers are the most mobile stage, first instar, second instar. So those are the stages that are more likely to move these pathogens around. That's why I think the vine mealybug has been more of a a danger, especially in vineyards, than some of these other pest species. Very quickly, we'll go through monitoring, chemical, biological controls, mating disruption, and I'll just talk about area-wide programs, not just for mating disruption, but for pesticide considerations as well. Briefly, monitoring, except for the Gill's mealybug, we have got pheromones developed for all of these different mealybug species. So you can monitor with pheromone traps for any of these insects that are really of interest to you. That would be the grape, obscure, longtail, and vine mealybugs. Not just using pheromone traps, but if you see ants going up and down the trunk of the tree or the vine, follow those ants. That usually means that there are mealybugs someplace, or scale insects. You can strip bark, but that's tedious. Mealybugs are usually in some hidden location. For pistachios, apples, pears, we oftentimes see the mealybugs on the trunk. In pistachio, really high development here. In vines, you can look for drop leaves. They tend to accumulate on the leaves, vine mealybugs, and that will drop the leaves. Grape mealybug, obscure mealybug tend not to get to such high populations. And for some of these pests, such as the Gill's mealybugs, going out at the springtime and looking at that budding area, sometimes that first generation, you'll find the mealybugs there on the buds. You could see it here with the Gill's mealybug and pistachio. I've seen the same thing when we've got about 4 inches of growth of vines. I go out there and look at those new fresh leaves, and that's where I'll find the grape mealybug populations developing. Chemical controls. It's going to be hard to kind of summarize chemical controls across all of these mealybugs, but what I'll point out is what I worry about, and that's the loss of the materials. So this was the 1990s when we first were looking at chemical controls for the vine mealybug. We were using Lorsban and Lanate. We looked for alternatives to Lorsban. We came up with Admire through the drip system, and an insect growth regulator applied in the summer against the first and second instars. Well, we know now we've basically lost Lorsban in most of our crops. And we've had both Applaud and the neonics questioned. Applaud has been on or off lists for vineyards and other crops. And we know now there's going to be changes to neonicotinoids. So we have to look at other materials. Spirotetramat, Movento, has looked as one of the better products. Other nicotinoids, such as Assail, are not on the directed neonic list, and they can be used as a knockdown. What I want to point out here, though, is we've looked, especially in vineyards, at all these different kinds of material. Movento has tended to be one of the better materials, but we have even seen some vineyard areas where material has been applied, Movento, and kind of broken down. So we're still looking at other products. We're looking at things like Savanto as a drip or as a foliar application. The thing to mention is that your PCA knows more about your vineyard, your apple orchard, your pear orchard than I do. And these materials have different efficacies depending on the crop, depending on the timing. So for example, using Applaud, we were always using it as a summertime material, which we could not use in vineyards. So we tested it as an alternative to Lorsban. And we can see here's the control. Here is Lorsban, and here's Applaud at 12 ounces and Applaud at 24 ounces. And we were able to get control into July with this delayed dormant March application without getting any impact on the fruit itself. So we're trying to retool some of these chemicals. Also to mention with products like Movento, again, it depends on your crop, because Movento, the active ingredient, Spirotetramat, has to be metabolized to an enol, and that depends on your crop physiology. So it may not work as well in pistachio, for example, as in vineyards, depending on how that pistachio tree picks up and incorporates the Movento into the enol product. Also to think about with something like Movento or some of the other neonicotinoids that are applied as a systemic, it takes a while to kill. Movento acts as a lipid biosynthesis inhibitor. So it doesn't kill your insect immediately. Sometimes people will apply Movento and a week later still see live mealybugs and they think, I've got to go in there with something else. It's because Movento takes a while to move to different parts of the tree canopy, of the vineyard canopy, and it has to impact the mealybug by having the mealybug feeding on it, and then its lipid biosynthesis stops working, and so it loses energy, and it's slowly starving out that mealybug. So for some of these products, you've got to give them time to actually work. I've also worked with a number of the OMRI materials with organic growers, and I know in North Coast, we've got a lot of organic growers and different crops. Most of these materials have a knockdown impact on the mealybugs, but most have not worked as well as the conventional materials. Part of this is because they've got a low residual impact, so you have to have repeated spray applications. They work better against the smaller stages. So if you've got overlapping generations, like with the vine mealybug, you have to have multiple sprays. And they can be broad spectrum, so they can knock out some of the biological controls. So very briefly on the biological controls, there are a number of predators that work against mealybugs. Green lacewings, some of the midges, our Mealybug Destroyer, that's the one you can purchase from a commercial insectary. But we've also got a lot of native beetles, such as Skimnus and Hyperaspis. That's the picture down here, a native beetle. And let's see if I can get that movie to play. Okay, so we're not going to have that movie play, but that's okay. One of the things to note is that they all have an impact by ants. So you've got ants, you've got to control the ants for the biocontrols to work. We've looked at trying to manipulate some of these things by releasing them through drones. So far, we've not seen a large impact on this. We're going to continue this work for another 2 years. I know in pistachio, David Haviland tested this and did not have much of an impact as well. We're looking at releasing parasitoids as well as predators to see that impact. There are a number of parasitoids that attack these different mealybugs. I'll just mention that these parasitoids tend to vary in their effectiveness depending on the mealybug species. The native grape mealybug has got a whole suite of parasitoids that attack it. They tend to be very effective at keeping our native grape mealybug at low densities. In contrast, the invasive Gill's mealybug, especially in crops like pistachio, tends to have almost no parasitism, and we get better impact from our predators. If you're interested, give me a ring and I'll go over parasitoids with your mealybug species and in your crop with you. I'm going to kind of conclude on this just talking about mating disruption. We've tested mating disruption out with all the various different forms. Typically, you're saturating the field with a sex pheromone to reduce or delay mating. We've looked at plastic dispensers, aerosol devices, sprayable, isomate devices. The one thing to note with all of these is that mating disruption works best when the population is low and when it's used over a larger area. And I'm going to give you some examples of that. We see here a female immature mealybug and a male immature mealybug. The male is more slender and longer. There is an adult male mealybug and a female mealybug starting to put out an ovisac and a male in copulation. For all the Pseudophycus species, they have to mate to put out a viable ovisac. What we see in this video here is a male mealybug attempting to mate with just a sliver of a septum. And in this trial, the male mealybug spent hours trying to mate. We've got a virgin female up here that it completely ignored. So what's happening is this is competition. The female mealybug puts out a sex pheromone, but when we use mating disruption, the lures, the dispensers put out so much more sex pheromone that the male mealybug can't find the female. This pheromone is being emitted. This is the hind coxa of the mealybug. The hind coxa's got these little pores on it, and that's where the pheromone is being emitted from. We know that, again, long-tail obscure, vine mealybug, and other species must mate to reproduce. They still can put out an ovisac if they're not mated, but that ovisac is unviable, and so no crawlers come out. Females can mate multiple times, but this does not increase egg production. And the one thing about the mating is that an unmated female will live for a very long time, up to 100 days, waiting to mate. And that's why it's important to have season-long mating disruption if you're going to do that. The males are very short-lived, the adult males. While they can mate multiple times, mating disruption will disrupt their activity so that they won't get to a female. They'll spend all their time and they'll probably die off on those false plumes. So I mentioned there are different impacts in the field that will have an impact on your mating disruption success. One of them is plot size. So what we see up here is one trial, and we've got this heat map, controls in yellow, mating disruptions in red, and the hotter the color, the more mealybug. So before this trial started, we've got a hot spot here, a hot spot here, and one developing up there. The wind direction is going from the north to the southeast. This is at the end of the first year of biocontrol. We can see that control still had some, mating disruption in the blue was less, and now we've had a shift, and upwind, we've got more mealybug. We did this for a second year, and again, we see upwind, more mealybug, the mating disruption fewer, the control sandwiched in between two mating disruption plots much lower, and at the end of the season, similar results, a little bit of a hot spot here upwind. So just remember, with mating disruption, it works better on an area-wide basis. And our damage was the same. We could see the damage up here. High populations, it doesn't work as well. This is just showing a low population, medium population, high population. A low population per capita, mating disruption had about a 90% reduction. High density had almost no reduction. That's because when the mealybug is clumped, the male emerges and just walks a few millimeters and finds the female. So it doesn't need to use mating disruption. So it works best in combination with insecticides. So just to conclude with area-wide, showing some data from one of our sites when we started the project in 2022. This is just a heat map showing all these hot spots with a lot of vine mealybug, starting off in 2023, monitoring, and we'll just see the development of these different populations of the vine mealybug. So what you can see is that working in an area-wide group, we were able to find the hot spots, work with those growers, let them know this is where insecticides could be applied, and using mating disruption, where needed, exactly like what they're doing with the Pierce's disease program you just heard. So in summary, California has a number of mealybug species, especially in vineyards, but also in orchards. They have similar biologies with a few slight differences, and their pest status to me comes down to their temperature development. More generations per year, like the vine mealybug, equals more damage, but they have different host plant preferences. There are natural enemies. Predators attack all of them, but parasitoids are the ones we typically want to use. Their species will vary amongst the different mealybug species, and their control will vary amongst the different mealybug species. No single pesticide is 100%, and they vary, but work with your PCA to see what works for you. I worry that we're losing a lot of these materials through either resistance or legislative rules. Mating disruption is there for the vine mealybug. It probably will be there soon for the grape mealybug, but it's not quite yet there. I like the idea of area-wide controls for all of these mealybug pests. Work with your neighbors, work with your county ag commissioner and farm advisors. Thanks.